welcome everyone. Today we have two very interesting talks, and our first one is going to be Arjun. And so Arjun recently joined Bogdan Pasaniuc's lab as well as Mike Gandel's lab as a postdoctoral researcher. He's done lots of really interesting work on TWAS and related topics in statistical genetics coming from Mike Love's group at UNC. And today he's going to tell us, I think, about one of his wrapping up PhD projects, if I'm correct. But anyway, I'll leave it to you, Arjun, and thanks for talking to us today.
Awesome. Yeah, thanks a lot, Harold. So Caroline asked me to start off with a little bit about my journey in science, which is a little ironic because I think it's just starting. But I don't have the most interesting of backgrounds. I've always been interested in math. So in undergrad, I declared as a statistics major basically on a whim while I was like being misguided as a pre-med. And during the grad school and med school interview process, I decided that I couldn't really leave my one true love. That's math. And I committed to staying in Chapel Hill for my PhD in biostatistics. I'm interested in developing methods that help decipher biological mechanisms from genome-wide and transcriptome-wide association studies. And that brings me to this work that I've carried over from grad school. I'm going to walk through some interesting genetic epidemiology type results in detecting gene trait associations specific to the placenta and the developmental origins of health and disease. In the process, I'll talk about some recent methods that I've developed to infer biological mechanisms from transcriptome-wide association studies. So one of the most important tissues in studying peri and neonatal health is the placenta, but it's often left out or underrepresented in large consortia like GTEx and ENCODE. Of course, the placenta is a very difficult tissue to harvest, so researchers need specific enrollment and harvesting plans to collect data for large enough cohorts. The Descartes project from Jason Dure and Cole Trapnell has taken us a step towards considering the placenta and other fetal tissues, but work on disease etiology is still underdeveloped. The placenta serves as the master regulator of the intrauterine environment through key biological mechanisms like nutrient transfer, metabolism, growth hormone production, and immunologic control. And all of these mechanisms have influences on postnatal health. Traditionally, the research paradigm here has been that environmental factors like chemical and physical, psychosocial stressors all affect the placental genomic landscape, but namely the epigenetic landscape. And that confers and effects on gene expression and other pathways in the placenta, and then in turn have effects on postnatal traits. So more recently, fetal germline genetics has shown some links to placental transcriptomics, and that relationship overlaps with some genetic signals for birth weight and childhood obesity. We also recently published a paper that showed that non-linearly aggregating transcriptomics and methylomics predicts social responsiveness and intellectual disability in the context of autism diagnosis. So all of these observations fall in line with the developmental origins of health and disease hypothesis, which suggests that the in utero experience has long-lasting impacts on non-communicable traits and disorders. So here we incorporated germline genetics and placental multiomics for a series of mediator-enriched transcriptome-wide association studies to address three overarching questions. One, which genes show associations between their placental genetically regulated expression and various traits across the life course. Two, which traits along the life course can be explained by placental genetically regulated expression in aggregate. And three, which transcription factors, microRNAs, and CpG sites potentially regulate trait-associated genes in the placenta. So I want to start off by talking a little bit about the methods we've used for this analysis, namely to incorporate genetic variants that are distal to a gene into TWAS. In expression quantitative trait loci or EQTL analyses, we often find these highly significant distal EQTLs where the SNP and the gene are far away from each other, but we usually lack some context as to why these associations pop up. For example, from earlier work in detecting EQTLs in bulk breast tumors, we found several of these distal EQTLs. They're represented by these dots off the diagonal on these cis-trans plots. Some of these distal EQTLs point to cell type heterogeneity, like the one that I've circled here, HGH1. It's a gene that's highly expressed in immune cells compared to other cell types in bulk tumors. But beyond reflecting cell type heterogeneity, these EQTLs contain information that could be relevant to trait regulation. Several studies have previously shown that transacting EQTLs often huddle around regulatory elements. So including these distal EQTLs that are actually mechanistically relevant in gene-based mapping techniques can provide some insight into gene regulation, especially any potentially tissue or cell type specific mechanisms. So this line of thinking is consistent with the Jonathan Pritchard Group's omnigenic model. The idea here is that gene networks are so interconnected that all genes in the transcriptome have some effect on complex phenotypes. Core genes directly affect the trait, while peripheral genes affect the core genes, and thus have these indirect effects on phenotype. But we can also move back into the central dogma and apply the omnigenic model to SNP gene interactions as well. Local SNPs affect genes through cis regulation, while distal SNPs have indirect effects on transcription through these transacting pathways. In the 2019 Cell paper that provides a lot more support for the omnigenic model for SNP to gene interactions, distal SNPs were estimated to explain up to 70% of heritable gene expression. TWAS has also incorporated regulatory information. For example, the EPIC-scan method showed that integrating epigenomic annotations into predictive cis-only TWAS models provides gains in prediction as well as TWAS power. So in this cartoon here, say we have these two SNPs, 1 and 2, that are both local to gene G. Traditional TWAS, like PredictScan or Fusion, is going to weight these two SNPs equally. Now, if we know that SNP2 falls in the promoter region of gene G, then EPIC-scan is going to upweight SNP2 and downweight SNP1 in the predictive model. These kinds of annotations are generally available from large consortia or databases, but they're limited when we're looking at understudied populations or understudied tissues like the placenta. So we set out to build on these lessons to use a data-driven approach to prioritize important distal genetic variants that are mediated through associated biomarkers into a TWAS framework. And we developed MostWAS, or the multi-omic strategies for TWAS. The software for MostWAS is available on GitHub as an R package, links right there on the screen. And the underlying machinery for the method is detailed in a recent PLOS Genetics paper with Mike Love and Yun Lee from UNC. I do want to go over an example biological mechanism that MostWAS leverages to train predictive models. So here in this cartoon, we have a gene G, transcript levels in blue up top. Far away in the genome, we have this distal SNP S in green that's found in the regulatory element of gene X. So let's assume that this SNP is regulating transcription of X, and it happens to be the fact that this gene X codes for a transcription factor that binds to the regulatory element of gene G. So indirectly, distal SNP S has this effect on a transcription of gene G through a transacting pathway. So if the association between the dosages between distal eSNP S and the expression of G is strong, we'll detect the distal EQTL. If this mechanism is actually true, then SNP-S has an indirect effect on the transcription of gene G, and we can call this distal EQTL a transacting EQTL. Accordingly, MostWAS can be used to detect similar distal regulation via differential chromatin or methylation states or microRNA binding. There are two methods for prediction in MostWAS. First, mediator-enriched TWAS, where we first detect the strong associations between the transcription factor X and gene G, work our way backwards to the SNPs around gene X. We can then build a local model to predict X and incorporate that into the final model for G using two-step regression. Second, we have distal EQTL prioritization via mediation analysis, where we can interrogate highly significant distal EQTLs like the one in this dotted gray line, to detect these large indirect effects on gene G that are mediated through sets of biomarkers that are local to the distal SNP. Distal SNPs with large mediation effects are included into the final model that we can fit using elastic net or linear mixed modeling. MostWAS selects the best model from these two methods as the final expression model, and then we can run TWAS by direct imputation or the weighted burden test. So that's enough for methods right now.
Can I ask you a quick question?
Sure.
So if you go back one slide, so how often do you have it where the regulatory SNP or the SNP that you have in the regulatory element for the transcription factor doesn't give you an EQTL signal for the transcription factor itself?
It's very likely that that's the case. So like that obviously is a limitation of this method, but we are mainly we're trying to restrict the multiple testing burden by searching for SNPs that are generally in these coding regions or are local to coding local to genes and also these methylation or epigenetic factors.
Mhm.
Cool. So I'm going to stop talking about methods for a second here and dive into the analyses. So our training set for this placental TWAS is from 272 samples from the extremely low gestational age newborn or ELGAN study. The co-PIs for this study are a couple of my collaborators from UNC, Rebecca Fry and Hudson Santos. The ELGAN study collected maternal and fetal placental genomics data from women at high risk of preterm delivery, and then tracked the children's physiological health and neurodevelopment longitudinally for over a decade. Here we're going to be focusing on the kids. We have germline genotypes from umbilical cord blood and gene expression, CpG DNA methylation, and microRNA expression from the fetal side chorion tissue of the placenta. So this tissue that we're looking at is mainly a mixture of trophoblasts, blood vessels, and mesenchymal stromal cells. So lots of proliferating cells like trophoblasts and these stromal cells. I'll be flashing this study scheme up every so often to guide us through the 4 steps of the analysis. And the first step here is going to be training expression models using MOSWAS by incorporating cis and trans SNPs. So out of approximately 12,000 genes with sufficient expression on our RNA-seq panel, we're able to build 2,994 significant gene models. And by significant, I mean that we can estimate SNP heritability of expression at greater than at a p-value cutoff, at a pretty stringent p-value cutoff. And our model can predict expression with at least a cross-validation adjusted R-squared of 0.01. We then subjected these models to external validation in a different EQTL dataset from the Rhode Island Child Health Study, where we found that over half of the genes that we could evaluate had R-squareds greater than 0.01, showing the distributions of in-sample and out-sample adjusted R-squared, with the mean and median marked as well. Of course, there's a drop in performance out-sample, possibly due to differing genetic architecture, but overall, the portability is on par with a lot of expression models from other tissues. So next we turn to the section of placenta-specific gene trait associations, or GTAs, by integrating these models with 40 different GWAS from European ancestry populations. They're all from large consortia like the EGG Consortium, the Early Growth Genetics Consortium, the Psychiatric Genomics Consortium, UK Biobank, GIANT, and the Complex Traits Genetics Lab. We first looked at if we can infer some kind of strong genetic architecture for these traits. We grouped our 40 traits into 5 general categories that have shown associations with fetal placental genomics in previous studies. In orange are the autoimmune diseases, purple gives us metabolic traits, in green are the cardiovascular traits and diseases, blue are the neonatal traits and outcomes, and in red are the neuropsychiatric traits and disorders. 37 of the 40 traits showed significant SNP heritability, and around half of them showed heritabilities of more than 0.1. When we scanned for genetic correlations between these traits, as expected, we found multiple strong correlations between traits of the same category. We found several, for example, this block here of purple traits, all the metabolic traits show strong correlations with the neuropsychiatric traits. But we also found several cross-category correlations as well, especially between metabolic and neuropsychiatric traits up here. These results aren't surprising, but overall, they justify a strong genetic architecture for a lot of these traits. Next, we added the layer of placental expression to these GWAS through TWAS. Here, we use the weighted burden test to scan for a gene trait association, and then use the permutation test by shuffling the SNP gene weights to test if the SNP to gene relationship tells us more than just the SNP to trait relationship from GWAS. So at transcriptome-wide significance, we detected 248 GTAs across 176 genes, with the majority of these associations coming from metabolic traits or neonatal outcomes. This bar graph here breaks down the number of GTAs per trait, and the orange portion of the bar shows the number of GTAs for each trait that have significant associations from the distal SNPs included into the MOSWAS model. I'll be expanding on these distal SNPs at the end of my talk. So in overrepresentation analysis for Panther pathways for these 176 genes, we saw enrichments for immune and cell growth pathways. These are all consistent with the morphology of the placenta. Placenta conveys a lot of the protective immunity from the mother, and since it's made of proliferating and growing trophoblasts, these cell growth pathways also make sense. In fact, there's a recent paper in Nature just this year, like this month actually, from Courns et al. That came to a similar observation about the proliferative nature of the placenta, that the copy number, mutational profiles, and the mosaicism in the placenta resembles that of a fast-growing tumor. So next we looked at the second study goal, about what traits we could explain by the aggregate of placental expression. We use ROGUE, an LG score regression-based method, to estimate genetic heritability and genetic correlations on the expression level. Briefly, we can grab the core equation from the LD score regression that links chi-square test statistics from the association studies to LD scores, and then cast this into a TWAS framework. So on the left-hand side, we'll have squared TWAS Z scores. This N sub T is our GWAS sample size. L is going to be the genetically regulated expression correlation scores. M is our number of genes, and then A is some controlling variables for population structure. So if we run this regression here, the coefficient of the second term is going to give us an estimate of the gene expression mediated heritability. So overall, with this analysis, we found that placental expression mediated heritability is higher for neonatal traits. So these blue, this blue box plot, and the three traits that we can actually find, we actually estimate to have significantly non-zero heritability are all neonatal as well. Childhood BMI, total growth in puberty, as well as the start of puberty. But when we looked at the genetic correlations on the expression level, we also found some interesting results. As expected, we found correlations between traits that are metabolically related, but also correlations between neuro and cardiovascular traits, as well as autoimmune and metabolic traits. So these correlations suggest shared genetic pathways for these pairs of traits, or for etiologic antecedents of these traits, either at the susceptibility genes themselves or through the shared distal loci. So next, we followed up on these gene trait associations with some computational methods to provide some context. First, we looked at many instances of potential horizontal pleiotropy, where one gene is showed associations with multiple traits. For example, we have IDI1. It's a cholesterol biosynthesis gene that showed positive associations with body fat percentage, HDL, fluid intelligence score, and negative associations with triglyceride levels and schizophrenia. So there's a link between cholesterol-related genes and schizophrenia, and that's been detected before, and that's potentially due to co-regulation of myelin-related genes. Another interesting thing about this is the predicted expression of IDA1 was built using distal SNPs found within the 12q24.31 locus. And that's a known risk locus for hypercholesteremia. So the inclusion of this region might have contributed to the large GWAS associations, but it also could point to another region that needs to be further studied for the co-regulation of these traits. Another example gene here is CMTM4. It's an angiogenesis regulator, and it showed evidence risk of intrauterine growth restriction due to involvement with endothelial vascularization. So if you let me be a little speculative here, this gene actually might illustrate an example of the DOHAT hypothesis. It's more likely that placental expression of CMTM4 has a direct effect on fetal birth weight just because of temporality, and fetal birth weight is positively correlated with body fat percentage and hypertension as measured in adults. So these results don't necessarily signify shared genetic associations mediated through placental expression, but they might point to complex genetic pathways within the placenta that have influences on antecedents of these complex later in life phenotypes. So we further studied this extension of our hypothesis by looking at 9 genes that showed 3 or more distinct gene trait associations across different categories. Using UK Biobank GWAS summary statistics, we conducted TWAS for a variety of traits across 8 groups, defined generally around ICD code blocks. Overall, many of these genes like ERP29, RPL6, RPS25 are all genes that code for fundamental proteins like ribosomal proteins. And as such, on the whole, we see that the gene trait associations in UK Biobank are enriched for immune or cardiovascular traits, as shown in these box plots of the negative log 10 p-values broken down by ICD code blocks. So if you look at the purple and teal here, these are our immune and circulatory ICD code blocks. More specifically, at the traits, we found a lot of associations with fundamental biomarkers like immune cell or blood cell counts and percentages. So this provides some more evidence that these potentially pleiotropic genes are associated with more fundamental Mendelian-like traits. We next examined whether placental genetically regulated expression of these 9 genes correlate with fundamental traits at birth. We imputed expression into individual level ALGAN genotypes and tested for associations for 6 representative traits that are either measured at birth or at 2 years. We have birth weight Z-scores, neonatal chronic lung disease, head circumference, fetal growth restriction, necrotizing enterocolitis, gastric diseases, and mental development index at 2 years. And overall, as shown in these forest plots of effect sizes and adjusted confidence intervals, we found negative associations between birth weight and SEC11A, head circumference, and ATPAF2. But these two associations here are particularly interesting. RPL6, again, is one of these ribosomal proteins, showed a negative association with this mental development index. And IDI1, as if you can remember back to the previous slide, showed associations with fluid intelligence as measured in adults. So as many of these genes encode for proteins involved in very core processes, understanding how the placental regulation of these genes affects neonatal traits can actually give some more context to the potential long-lasting impacts of placental dysregulation. But again, this would probably require larger longitudinal studies that involve genetics. So I'm going to finish on a unique flavor of MOSWAS, and that's the ability to generate functional hypotheses for transregulation of TWAS identified genes. I'll be focusing mainly on transcription factor encoding genes in these slides, but we do find that many of the mediating CPGs overlap with placenta-specific cis-regulatory elements. 89 of the 248 GTAs showed significant associations through these distal loci. I'm listing 3 example transcription factors here, all of which have literature support for placental regulation, but I want to hone in on EPS15. EPS15 is a transcription factor that's maternally imprinted and has previously shown to be associated with fetal growth restriction. In our TWAS, we found that EPS15 negatively is associated with SPATA13 and FAM214A, and all 3 of these genes show associations with waist-hip ratio. So we then turn to the Rhode Island Child Health Study again to computationally validate any of these transcription factor to TWAS gene pairs detected in LGAN. We use the GBAT method to detect a transgenetic association between the locus around the transcription factor and the expression of the TWAS gene, as well as agar regression-based Mendelian randomization to estimate a causal effect in this direction. These forest plots give the GBAT and MR effect sizes and adjusted confidence intervals, showing further evidence here that EPS15, there's this directional association from EPS15 to FAM214A, particularly that we can predict EPS15 to downregulate FAM214A. So to tie off this whole story, we took EPS15 and its 2 targets to in vitro experiments so we can do a 2-for-1 in vitro study. So using human placenta-derived trophoblast, we used a FANA-silencing RNA to suppress EPS15 expression. As a negative control, we used a scramble oligo to shuffle the sequence of the EPS15 silencing RNA. And then we measured expression of EPS15 and the 2 targets with qRT-PCR. So this box plot here shows the fold changes of these 3 genes across our 3 groups. In the FANA knockdown, we see that EPS15 is successfully knocked down, and that SPATA13 and FAM214A showed increased expression in these knockdown cells. So this provides some experimental evidence of the inverse relationship between EPS15 and these 2 targets that we computationally predicted. Of course, we haven't investigated the gene trait associations themselves, but the gene functions point to the proliferative processes of the trophoblast layer of the placenta. EPS15 is a transcription factor in the EGFR pathway, involved in cell growth functions, and has known roles in endocytosis. SPATA13 has actually been implicated as an oncogenic gene with functions in cell adhesion and proliferation. Fortunately, FAM214A's function hasn't been categorized yet, especially in the placenta, but the conserved protein products of the locus are all involved in chromosome segregation. So all 3 of these genes share functions that are key in proliferating cells, like trophoblasts that make up the majority of the placental tissue. So in conclusion, first from a methodological perspective, our analysis shows the utility of MOSWAS in generating functional hypotheses for tissue-specific upstream regulation of GTAs. And in this one instance, our hypothesis actually holds up to experimental rigor. Our analysis also adds a layer of genetic influence to the DOHAT hypothesis. We showed that placental expression in aggregate is more likely to explain significant portions of early in life traits, but on the margins, several genes show associations with traits all across the life course. The genes that showed associations with multiple traits across categories tend to correlate with the fundamental biomarkers. So our work here offers evidence for a modification of the DOHAT hypothesis that dysregulation of placental genomic regulation is more likely to have direct effects on fundamental fetal or neonatal traits, and these fundamental effects compound and manifest in long-lasting impacts on health. On a more macro level, our work also underscores how much work is left to do in studying genetic effects on neonatal traits. And we emphasize larger GWAS cohorts for early in life traits and the placenta as well in tissue-wide regulation studies. So this project has been a really massive effort with lots of data sharing, lots of collaborators around the world. So here's a list of them all. I also want to thank Bogdan and Mike for having me in for this opportunity at UCLA. And I'll take any questions and comments right now. Thanks.